It's an hour-long segment where I go over some of the products, tools, tips, ideas, workings, status reports, all that stuff associated with innovative potential. Eventually, we're going to get to some gaming, but if you have any questions or comments in the chat, you're free to join in Tuesdays at 10 for the segment. This week, we're going to be doing the cancer biology wheel. It's available for 99 cents on the Innovative Potential website, theinnovativepotential.com. And this is intended as an oversight or overview of basically cancer biology in a nutshell. So if you're new to the subject, if you're new to cancer, study sessions, if you're in med school kind of thing, this is, I developed this tool myself to get a handle on what the hell was going on. The segment's a little bit more candid and it's a little more flexible. And we're going to just sort of go real fast around the outside. And just sort of more or less walk through the stages and steps of cancer initiation. And so how this kind of works is all of this is based on Hanahan and inspired on a paper done in 2011 by Hanahan and Weinberg, and it was it was the first paper, one of the first papers to attempt to put everything in one picture. And this paper was the Hallmarks of Cancer. And since then, there have just been, and this was done in there was one paper done in 2000 and another one done in 2011. And since then, there's just been this flourishing of work, of microbiology. It's been an explosion in the sciences where it's like a lot of stuff got known, a lot of stuff got introduced, a lot of new concepts, a lot of mechanistic, behavioral stuff in terms of cancer biology and tumor development really kind of got more understood. Basically, in a nutshell, the way I kind of broke it down is there are 4 different segments or different key type of information chunks or strategies because cancer is like a whole body disease, so it goes from the genetics to the cell to the tissue. It's sort of an all-encompassing disease. It's kind of why this is going to sound strange, but it's kind of why I like it. It's very interesting. It's really something you can dive into and get a handle on from almost any general direction in the life sciences, and that's sort of what this image here on the screen is attempting to convey, that you can actually approach this subject from any of the sciences. But cancer biology for me and the philosophies at Innovative Potential are basically, there's the genetic component of the disease, there is the cellular component of the disease, the tissue component of the disease, and the environmental or inter-organ environment component of the disease. And most people started to get introduced to it, the subject, where it's like, oh, okay, if it's not a death in the family or something like that, people get introduced to cancer in their pathology courses, and I'm speaking more at the biochemical level, or in cancer biology courses. But it's all just cell cycle. So you can actually start being introduced biochemically to this somewhere in your high school biology courses. Where this goes, There are, first thing I want to make mention of, there are a lot of terms on this. There are 24 terms and concepts of how a tumor, how a cancer gets a mutation, expresses changes in its DNA, different phenomenon that are happening within the cells because of that mutation, and you're just sort of spiraling downwards between this this initial mutation initiation, and then here comes this big sort of series and comedies of errors that just allows this cell to just sort of proliferate out of control and grow tumors, basically. I don't use the word metastasis here. It's implied, and I'll get to that at the end, where it fits. But this is intended to really get a handle on when new studies come out, to sort of pinpoint what types of signaling pathways or phenomenons are happening where, and what they actually looked at, and trying to get a handle on it. And this is the big, and this is the big wheel here, because it's like the colors are actually significant. There are biochemical pathways between each of the colors. So between the xenobiotic exposures in red and the cyclin-dependent kinase overexpression, also in red, that is cyclin E-dependent nicotine expression. So you inhale the nicotine acts on its own pathway that increases the level of cyclin. And cyclin E does this thing where it increases the frequency of cellular replication. So there are actual deeply rooted cellular pathways going from the internal color section to the external color arrow. And I tried to set this up on purpose because when you start looking at when you start really digging into the molecular and cellular biology stuff, you get a really good idea of how this disease actually functions in terms of it's a very big type of retrograde, where the rest of the cells is the normal cell, normal functions. But as you see cancer and tumors progress, you really get an idea of no. This one cell with the mutation is actually losing functionality. It's losing definition and becoming a cancer. So I'm going to start with this at the local microenvironment feedback, and we're going to just follow the wheel going in and out into xenobiotic exposures, altered gene expression, genetic mutations, P53 aberration. There's genomic instability, Warburg Metabolism. I'm not really gonna list them off, just for the sake of listing them off screen. Basically, at the local microenvironment feedback level, this is something I'm going to use smoking because smoking is one, but it can be something like I believe it's Helicobacter pylori, ulcer causes, chronic alcohol use. It can be there's so many different factors. It could be the sun. We can use the sun for this one. So basically, you're in a tanning bed. Or you're smoking and you're in a tanning bed, both at the same time, kind of building a little bit of straw man, but you can kind of begin to develop your own understanding of how this all actually just fits in together. So the xenobiotic exposures are different chemicals or something like that you're going to pick up So I'm going from the local microenvironment feedback to xenobiotic exposures. I'm going from something mental outside down into right into the tissue level. So right down into the histology kind of microscopic areas and the biochemistry. So there's a big collapse right kind of drawing in and how you as a person interact with the environment, and how that environment then interacts with your tissue and the cells, and really Magic School Bus here, where you're just shrinking down smaller. Fantastic Voyage, Magic School Bus, you're just shrinking in smaller and smaller and smaller. Kind of wish there was a video game like that, because I'd play the shit out of it. But there's really nothing on the market that's doing a Fantastic Voyage right now. So bam, let's get that up and running. But anyway, so the xenobiotic exposures can be anything. Basically, xenobiotic just means it's not of the body, it's not biological. This was typically developed in this language is typically developed for chemicals, but it can be expanded to just sort of mean like radiation or UV light or anything that's going to be a causative agent. Alcohol can be a xenobiotic exposure kind of thing. Basically, some of these chemicals do affect gene expression, particularly certain types of hormones. So the idea here is to sort of build your can in your head yourself, because this is how it happens. You get these environmental factors that then sort of get in a cell that may not change its behavior because of its exposure. Between altered gene expression and genetic mutation, they play off of each other. They're interrelated, because an altered gene expression may or may not can be upregulated, or it can be downregulated, or it can have more of the protein product of the gene created for you. If that increase or change has a mutation in it that then codes for something that is non-functional, typically it's like a wash point. But sometimes in the regulatory sections of the protein expression, you can have you can go to a situation where the gene itself is just permanently expressed because of the genetic mutation. And that's kind of what I mean. You kind of get into these hormonal types of things with like aromatic hydrocarbons and the ARX, arohydral receptors. This is sort of where food and don't eat charred meat kind of thing, like permanently and chronic kind of stuff that kind of comes into play. Don't smoke. That's where this type of happens. It's like you get exposure, but then your body adapts to that exposure in some way, shape, or form to help restore the body to homeostasis. But if you end up with a mutation in some of the regulatory pathways of that homeostasis complex, that's when you start to progress on your road to cancer, and that's sort of what the big thing here is. Congratulations, you got exposed to something, could just be the sun or whatever. You ended up with a mutation. Sometimes the mutations just happen, and that's what I want to stress here, is like sometimes mutations actually just happen as shit luck. The whole, you're going through like a mole worth of DNA base pairs at any given time, in your body, replicating DNA on its normal functioning. Sometimes just the mutations, just like it's just plumb shit luck. You go through these stupid base pairs. Something's going to happen. It's not absolutely perfect. It's damn good, and there are some balances, but there are, something happens. Sometimes it's no fault whatsoever. It just kind of happens. But we do know a lot of things, particularly like that cause these types of like mutations and stuff to like really promote them, kind of thing. But you end up with genetic mutations, and you can have these genomic instabilities. What genomic instability is, it's something like you can have your whole chromosome sort of have these like a chunk of your chromosome break off into another chromosome and have it like that ends up creating some of the childhood cancers and stuff like that. I don't dive too deep into that because it really isn't to an hour is insane. But really look at what Google these terms and put it in Google Scholar and PubMed, and you just get this huge, you get like decades worth of definitions. You get decades worth of high-end scientists just like these are like major career papers for a lot of people. So I'm not going to explain today. I just want to make people aware that this word exists. You can get information on it. There are Wikipedia articles if you're not to read journal articles, but can start to get a handle on what really is what you're made of, kind of thing. P53 aberration, and this is an important one because it's a major cell cycle suppressor, and it's basically if there's going to be something wrong, it's like the P53 is to a dividing cell. So when the cell divides, it divides, but division is carrying a mutation that promotes additional divisions. That's how we start getting into problems. But anyway, all of this sort of working through this cascade at the genetic level and how chemicals interact with genes, and then how those genes go into expression and cell cycle regulation. That's huge. That's one of those like unending. There's multiple subspecialties just for P50, just to be able to study P53 aberration. Basically, why I pulled that out is because if there's a mutation in the P53, it's basically you're not able to stop your cell cycle well enough. So if there's a problem with this gene, with this protein product, either the gene gets cut off or there's a defect in the expression of the protein itself, then you've set the stage for this cell to be able to create a cancer. It hasn't happened yet, so we're always sort of starting with like, here's the next step, here's the next step, here's the next step, to try and get a better understanding of where these drug target locations are, where can we stick extra treatments, where can we actually make significant gains with all of this like Human Genome Project level sophistication and understanding. I draw attention to Warburg Metabolism because this is one of the major components, and there's a lot of work done in terms of how altered. Basically, by the time Warburg Metabolism is observed, the cell itself has changed its basic aerobic components to favor phosphorylation and phosphorus use because it's making extra DNA. So when the cell is kind of stuck in this let's make DNA replicate forever kind of thing, it loses loses its capacity. It doesn't lose its capacity. It shies away. It starts moving away from aerobic respiration and starts to favor glycolysis because you're getting to these reactive oxygen species. And the basic component of this is that the DNA replicating in a reactive oxygen species rich environment is counterintuitive for the purposes of replicating DNA. So if you shut off reactive oxygen species production through mitochondrial respiration and that higher energy level, then you end up at glycolysis and it's lower oxygen demand, which then favors accurate replication of the DNA. And there's a couple of studies out there that look at phosphorus demand and glycolysis in terms of cancer and how they're done. And the Warburg Metabolism is basically a normal a cell. I don't want to say normal, but it's basically a normal cell going into this glycolytic pathway to favor, for the purposes of a safe space DNA replication kind of thing. That's a very important example of altered cellular metabolism. So already we've kind of gone from this molecule has made a DNA mutation. The DNA mutation has kind of like shut off one of the safeguards of the cell, and now the cell is just sort of left in this like unregulated market free fall kind of thing, where it's just sort of like spiraling. It still functions. It's still doing its thing. It's still, metabolically capable, but it's still going to just sort of like, it's unhinged. It's a little bit, it's not quite, I don't want to say it's unhinged yet, because it is still tightly controlled, but it's changed what it's doing. It's now starting to favor a pro-replicative environment. And this kind of opens up what's happening at the cellular level, because at the genetic level, the cell is just understanding, here's the instructions, I'm just going to do the instruction as it is, not recognizing that there is this mutation. So it has a blind eye. So at altered cellular metabolism, you start seeing these shifts to start favoring a pro-cancerous state. And at that point, you start to change the cellular identity. What this means is, and this is tied into cadherin and major histocompatibility complex changes. This is the idea. All normal cells have a certain identity associated with them. They freely express it. It's like they wear like little name tags that are protein structures expressed on the surface of the cell. And they have little markers that sort of attach them to their adjacent cells and to the basement membranes that they're attached to. It's like being in a bar or a crowd where you're just kind of there, but you all have eggs and you all have your job. You got your job, so that everybody in the room kind of knows, okay, you do this, and it's a very coordinated, extremely coordinated system. What starts to happen now at the cancer cell, because it's now switching into sort of like a survival mode. It doesn't necessarily have to express its name tag anymore, because it's more concerned about doing something else. Replicating, for example. So as long as the cell is able to replicate, then that's fine. But these are some of the changes that are made to cancerous cells as they sort of develop. So we're moving from a loss of cellular identity into increased frequency of replication. So this transition, it ends up with and this is all still part of your G and S phases, mitosis and meiosis cell cycle structure stuff. But what's happening here is, typically in a lot of cancers, you end up with cyclin-dependent kinase overexpression. And I focus this one out specifically because of that nicotine xenobiotic exposure type of upregulation or altered regulation of the basic core cell division apparatus. So that's really kind of what I'm focusing on here. And depending on where you're coming from, if you're coming from, and I'm talking in terms of the sciences here, if you're coming from molecular and cellular biology, some of this stuff you might already be familiar with, but as you enter into the oncological world and as you enter into cancer biology, these start popping. These cell cycle aberrations start popping up, and depending on when you get your cancer information, so is this a tumor that's been growing for 10 years, or is this a new initiated in silico lab on a chip type of, no, we created a single point mutation here and then we watch the after effects. So you can come from any direction on this, but basically in the generalized case, most cancers usually have some type of cyclin-dependent kinase overexpression, and these are the molecules that basically grow a cell, split a cell, rinse and repeat kind of thing. If they're overexpressed because its suppressor, the p53, isn't working anymore, then you're headed for a tumor. So basically, this little straw man of a wheel here, like hasn't really turned into a full-fledged cancer yet, because the cell may or may not have its apoptosis machinery in place. And the apoptosis checkpoint is gigantically huge because it's one of those essential to life checkpoint. It's essential to life to control this type of stuff. So what happens here is the cell suicides. The cell is able to not replicate or be alive anymore. And this is sort of the big driving factor if you're going to end up with a cancer or not, because from this point on, if you fail your apoptosis checkpoint as a developing sort of disease, then you can start ending up with clinical presentations, or you can start understanding or seeing clinical presentations. And I wish I could talk more about the apoptosis checkpoint, but it's one of those things where it's complicated. It's its own show basically, and I don't have time. But basically, if there is a mutation, or if there is something wrong with these proteins, these complexes, then the rest of this wheel is backing up. So basically, you're heading into overcrowding with an increased frequency of replication. You're going to start to begin to densely populate the local region, and that's not what you want, because it's like that old Simpsons joke with the feast of maximum occupancy. You have back to the bar example where everybody's got their like name tags on, their name and the job or whatever. Suddenly 5, 6, 7 extra people came into the building. you're kind of over your fire code violation, but nobody's really paying attention because maybe it's just that your name tag fell off, so the security isn't really worried. Like, they're doing their job, but they're not realize that somebody just sort of teleported into the bar, basically. That's kind of the analogy that works here. So now you're over capacity in your bar, and this is where you have to sort of zoom out from the genetic level, from the cellular machinery level. You're going to zoom back out again from the cell and its adjoining neighbor level, and now you're kind of getting into at your stage 1, stage 0, stage 1 carcinoma in situ type of microscopic histologic understanding of sort of what's happening here. So basically, with a little bit of overcrowding, there's now limited resources, and the localized region is now suffering from oxygen deprivation. But because the cells themselves, the cancerous cells themselves, have already switched over to a glycolytic pathway way back at the Warburg Metabolism there, it's more that the adjacent cells are starving for nutrients. So that's where this sort of happens that's why transient nutrient supply comes up next. It's one of those things where it's like, if everybody in your bar is now hungry, but we only planned for 50 people, but somehow we have 70 people, wedding crashers a little bit, we don't have enough food for everybody. So it's like, okay, well, let's order more food. That's the cytokine signaling. So at this tissue level, you're starting to get into distribution type of restructuring. And I'm going through this faster than I want to. Because I'm going through this faster than I want to. So basically, what's back to overcrowding. The overcrowding builds up to tissue stress, and along the way, the overcrowding, the cancer cells don't care because they're using glycolytic pathways. They don't need oxygen, but in the same way a normal cell does. But at the same time, the tumor hypoxia calls in additional resources, and this ultimately turns into a call for angiogenesis or additional blood vessels penetrating into the hypoxic region because of the overcrowding, because the cells are replicating faster than the food, material, resources, nutrients can access them. So this transient nutrient supply it's like a wound at this point. It's like a scar. It's like when you cut yourself, or when you get I'm figuring in the metabolic pathway at this point, where it's like we've entered into this wound healing response phenomenon for basically the last third of the wheel. This is all wound healing response type of behavior, where there has been a change there's a change occurring at the tissue level. The tissue embedded lymphocytes, the tissue embedded macrophages are still looking around, trying to see, like, well, who pulled the fire alarm in the bar, basically, or why isn't there enough food at this region? So I'm really just trying to paint this picture of where and how and why this godforsaken thing just ends up becoming a clusterfuck. And it becomes a clusterfuck because it's already a clusterfuck. Because when you start to, the way I understand it is, this thing is cancer is olfactory driven. There's an interplay between these bastardized cells and a normally functioning immune system, but the immune system doesn't have any molecules to base its discovery of the cancerous cells on. And that happens way back at the cadherin and major histocompatibility complex changes. So what happens is these white blood cells are just sort of looking around, making sure everybody's got their name tags on in the bar. It's like, are you over 18? And you put the little X thing on the, are you over 18 or can you drink at the bar? Like, it's an all ages night, so it's like very European kind of thing. But that's the mix and mingle that's going on here. So it's like, basically, you're getting underagers in your bar or something like that. That's sort of the imagery I want to provide here, because it's not a fault. No person or nobody's at fault. It's just there, but it's incredibly difficult to get a handle on the disease once it's sort of hit this point, because the system that is supposed to be in charge of stopping this phenomenon from happening is blind. It's blinded. It can't smell. So it's basically, if you have a police sniffing dog that can't smell anything, the dog is not really doing its job. You still bring it around, but it can't smell the drugs. It can't find the thing that it's supposed to do. It almost has no purpose. Like, you know what I mean? Like, these white blood cells can't smell what's happening. So this tumor cell is creating an environment and is setting off these not so much warnings. Yeah, they're maybe not warning signs, but they're creating a chemical signal that says, no, pay attention, I'm doing something wrong. But the thing that's supposed to stop it is not stopping it, because it's like a blind police officer. A deaf, dumb, and blind police officer with a dog that can't smell. So it's the cancer is like wearing a cloaking device. It can't communicate that something's wrong with it, and vice versa. The external regulation isn't there to shut it down. So you get this double negative where everyone is going to lose, because way back at the beginning, this cell got exposed to something or made a mistake somewhere that, hey, it's on its slippery slope. So back to overcrowding and tumor hypoxia and this transient nutrient supply. This is a situation where, if left unchecked, this is where you're going to start getting new mutations within the same tumor. So it's not just the one. If left long enough, you make more mistakes and you make more mutations. So it's at the overcrowding, tumor, hypoxia, transient nutrient supply section that you're going to start getting these the heterogeneous population of like multiple different cancer phenotypes developing within the same tumor. So this is one of those, like, if you end up with like a fist-sized tumor someplace, yeah, it's not just one type of cancer in it. There's like 3 or 4 different types, and they're going to have different adaptation strategies to them because they're in this constant like extreme, and they're in this consistently in this extreme environment. They're getting really aggressive there. It would be like you as a person not breathing for and just holding your breath randomly just to build up your ability to not breathe, or like that's sort of the extremely stressful environment from the cell's perspective that it kind of got forced into way back at the genetic level on this wheel, way back when it started switching over at its genetic mutation. When it got its genetic mutation, it started getting more and more sort of primordial in its behavior so that it can survive in this like bizarre, almost bacterial environment here. So anyway, this little micro environment here starts producing different signals to say, wake up, pay attention. And all of that sort of the tissue stress, because at this point, there are different interactions with the cellular membranes, the extracellular matrix. There can be remodeling at this point. This is where you're starting to see, it's like, okay, no, we're getting loss of borders here. We're getting penetrations. We're getting new blood vessels at angiogenesis. So by now, the rest of the organ system has started, okay, no, we're going to start, okay, relax. We're going to fix this. And all of this is mediated through the wound healing pathways. They're your regular wound healing response, and body development, cellular, molecular bio pathways. Meanwhile, there is this additional recruitment for white blood cells, and this is your inflammation response. Inflammation can start to develop depending on the severity of tumor hypoxia way back. So like this is how I really have to emphasize, take a look and understand what each of these key phrases and these key concepts, they're just whole concepts. They're not even words. They're like whole biology degrees in and of themselves, just stacked next to each other. And when you start to really understand how they overlap and feed and play off each other, then you really see the interplay here. And it's just like this, like it's like a woven carpet. It's like a woven tapestry of like, here's this, here's this thread and here's this other thread. So if this whole piece is responsible for one type of protein that is going to help me identify myself against the white blood cell, and it's gone, or it's been modified, took the lid off, it's been modified, but it's still interwoven and cross-linked with this one, and it's like the threads in your sweater or in your clothing or in your pants. This really is the complexity of it, because now it's like traditional drug development saying, okay, let's find one molecule that does something and then we'll just give it to someone. And that's where I'm coming from with innovative potential. That it's just like, that approach is kind of exhausted. We can't be doing that anymore. Because you end up with these, for the same reason there's a transient nutrient supply and tumor hypoxia, there's not enough oxygen getting into the tumor. It's the same reason why your drug is failing. It's the same reason why there are these increased expression of drug efflux pumps. This is the whole fucking show here. And it's its own thing. It's its own beast. It's its own species. Where I'm coming from right now is when the tumors get to a certain scale of volume, they end up expressing like a positive osmotic force. So basically, it's pumping stuff out and away from it, and it's creating its own little cancerous environment there. And that environment is usually necrotic. So the tumor itself is sort of like a hollowed out shell of just like pus and blood, and what's sort of left is like this like tumor basically. It is not a functioning healthy organ. But that's your number one foe in drug development, is how do you overcome this? So all of your other organs, all of your other tissues they're uptaking nutrients from the environment. They work normally. It's like houses in your neighborhood with the mailman. All of the houses work to have the mail come in, every day, and the newspaper, and like it works normal. It's on its good healthy cycle. But then one of the neighbors, put trash all over their yard, and one of the neighbors, and then that same neighbor, parked a car on their lawn, and another neighbor put, they're making, then they got the dogs. So it's like they made access to their house for the mail carrier more difficult. That's a little bit Stepford and suburbia there, but the same thing applies in residences. The same thing applies in shared housing. The same thing applies in apartment buildings, where something has changed and it gets it harder to fulfill normal functions. The delivery man can't deliver your pizza because your dog is like barking behind the door, or whatever. The point is, this tumor, this cancer cell, is working in the polar opposite direction of everything else around it, and is pushing stuff away from it from the inside out. The tumor pushes the drugs away from the interior of the tumor. That's how drug resistance develops, because of this tumor, this transient nutrient supply. If I can't get nutrients in there, and there's all of these like bastardized type of capillary vessels and blood flow, how does your drug get into the tumor when oxygen can't even get into it, and your entire body and billion-year history runs off of oxygen getting into the cells? So you got to work around that, and that's where your resistance is developed. That's where like big problem number one is the biggest problem to the treatment of the disease is the disease itself. That's where you got to really start using your brain here, as this like, That's where I like intratumor injection. I love intratumor injection. It just sort of cuts out the middleman. You can do what you want. Unfortunately, it took decades to get the infrastructure and the CAT scans and that type of stuff to the point where it's like, okay, at least we can at least begin to see it as a viable option. That didn't exist in the 50s. That didn't really exist in the 60s. It kind of came about in the 70s, 80s, a little bit more in the 90s, but we're only getting up to the point now, today in 2021, where it's like, no, yeah, we can kind of say in semi-rural communities that, you can get a CAT scan and we can start working out some of these solutions. That's a tie-in and a back out, and I want to go back up to into the pink there with inflammation. The inflammation is there to try and clean up these the tumor. So these macrophages, these white blood cells, go into the tumor and they start doing their thing, but they they end up they're still blind. So it's still the blind man with the blind dog, or the dog who can't smell. They kind of just keep adding fuel to the fire, making it worse. You're now dealing with this wound. You're dealing with a wound that can't heal, because the source of the wound isn't a cut or a scrape or a scratch or a chemical or a bacteria or a virus or something. It's the actual cell itself. So you gotta go way back, or you gotta go back to start. But there's nothing inside the cell with the mutation to shut that cell off, unless you pop the bubble with a pin, unless you apply a drug that will burst that bubble. Don't try and shut the DNA down. Burst the cell. That was the whole approach behind electroactivated chemotherapy, because it's like, if you can, and some of the nanoparticle stuff, it's like, if you can get your drug into the cell, you don't want to do that. But this is like one of those compassionate care systems where it's like, this disease has gotten away on you. You got to do something. You can't cut it out. You can't do stuff. So just stop everything. Full stop. It's kind of where that approach is where radiation comes in. Is if you can just overdose and zap a localized section, then you're good. You're not good. But you can make an extra step of You can make an extra step of progress, and that's really kind of what the purpose is at the clinical stuff. That's where at the experimental level you can start looking at, not clinical level, nowhere near clinical level, but you can start looking at different types of mechanisms that are going to either gently lyse cells in it'll cause necrosis. Yes, there will be problems, but with intratumor injection, you get a whole bunch of new drugs to use. You get a brand new bookcase of drugs that can never be used intravenously or inhalantly or through the diet. You can't eat the pill because it'll fuck your liver over. But if you use an intratumor injection and just inject the tumor, this is easy. This is a hell of a lot easier said than done. Okay, no ifs and qualms about it, but if you can stop that wound that can't heal itself, now you can start looking at solutions from regenerative medicine or plastic surgery, or you can get your control. You can get your control back, and that's sort of what this is all about. It's like when you truly lose control, we're on the cusp of getting another round of control when we lose control. So it's important to understand why, when, and how you use this technology. When you do have about like 3 or 4 different frontline, there's 3 or 4 different lines of treatment already, but people still die from the disease. This is an explanation of sort of how and what can be done at the later stages when the other ones fail. As you know more about it, you can move those backstage treatments a little bit closer to the front, and that's where cancer biology is going. That's where clinical treatment is going. How do we just can we get intratumor injection on the front line? Congratulations, we found your tumor. Can we just zap it? Not with radiation, but with can we actually get we have our treatments, we have our cures, but can we get our panaceas? We're kind of on the cusp of the panaceas of all-cause general cancer shutdowns. So anyway, back to inflammation on the pink there. The inflammation and cellular proliferation in terms of the cancer is basically, if you're going to have a cancerous cell start to move, start to be able to multiply, get away, and just sort of go off in any direction and find a new tissue to sort of colonize, this is when you're going to start to get it, because that's what the epithelial mesenchymal transition sort of is. It is a changing of the cell, of a cell type into another cell type, and replication. That's where the cellular proliferation comes in. So it's all kind of happening at once in an attempt to heal this wound that never heals. And again, depends on where you want to go. If you want to go down to the genetics of the EMT transition, the EMT there, or cellular division or cellular proliferation, you can. If you just want to, this is the thing I'm saying is that all of this is known. What order it's known for your individual specific case, a lot of times that really kind of doesn't matter, unless that's sort of the PhD level there, where you're into the thick of things, into what signal switching is signal switching when to make this happen. So basically the tumor environment boils over and reaches a point where it launches off a cell, and that cell goes off to divide, and it's migrating, and there's a full set of biochemistry again, and I'm being cheap. I'm not using the matrix metalloproteinases and getting into how the moving cancerous cell grows these little I want to say polyploidy fleet, but they're little like a mollusk has that foot. They grow these little feet, and then they just sort of scuttle off, kind of thing. There's real, go on YouTube, look at how it's done. There's beautiful stuff, but basically, you get to this point in this development, in this biological journey, where you start to come full circle, and you end up with the M word. This is where I used the M word. I wanted to, way back at the beginning, said I wasn't going to use the word metastasis. This is where metastasis happens. This is the process of metastasis from cellular proliferation, cellular migration. And that's basically what an injury-induced stem cell niche is. It is a metastasis. It is a cell that has gained enough capacity, surviving its own destruction, to be able to walk off independently of its own and create another colony of itself. Just like there's our stem cells, our cancer stem cells. This is the mentality and the mindset that you have to be in to be able to solve drug-related development problems. It's injury induced because there is still a mutation happening somewhere in this metastatic cancer. But the worst part is it's not just a single metastatic mutation, because by the time you get up to this cellular proliferation and migration and generalized metastasis, there are multiple cancers being spawned from the same tumor. You can have different flavors of cancer being spawned from the initial mutation. And you want to use a single molecule to shut the whole thing down. No. That's the part where I'm pissed. And then that's sort of where that's where people die. That type of, hey, if you're in med school, if you're in research school, if you're suffering with some of the diseases, if you're the doctors in clinic, this is sort of that palliative care interface, where it's like, how do you get people how do you give people in palliative care extra time, but good, meaningful time? Or in late stage disease, good, meaningful time. And this is sort of where my whole sort of professional life exists. And how do you get extra time to this sort of this is the Hail Mary play, because you want to kind of be able to answer, why did metastasis either enter into a type of hibernation and just sort of sit there, and that's been known to happen, where the single snowflake comes off and just sort of waits and just sort of stays there. It could be months, and it could be sometimes years, where it just sort of exists, not replicating, but then something switches it back on. Something in its local microenvironment switches it back on, and the whole thing starts over again, like a bacteria. So that's where the injury-induced stem cell niche is coming from. Because somewhere in that cell, there is still an apoptosis checkpoint pathway. So it might be metastatic, it might be aggressive, it might be that single cell that's just, hell spawned. But the hope is that, can you find that apoptosis checkpoint, and can you find that needle in the haystack? That's what a lot of people are working on. But that's not ignoring the rest of the bastardized tissue level stuff. So kind of what happens there, so once you get up to that injury-induced stem cell niche, that metastatic cancer that went off on its own and started creating another overcrowding scenario with tumor, hypoxia, and the injured cell goes off and creates its own toxic environment again, ignoring its own checkpoint and doing the whole thing over again. And that's cancer eventually, and death occurs when these tumors, this bastardized this tissue environment, overwhelms the proper functioning of the organ itself. So you can live with tumors without You can have cancer and not know it. You can have tumors and not know it. You can be asymptomatic for years or decades. Because this is a wound healing process, sometimes the body just self heals. It's one of those things where there's a lot of hope, but there's a lot of knowledge that has to be known and learned to be able to make any progress. And that's the cancer biology study wheel from Innovative Potential. It's available on the Innovative Potential website, and it's 99 cents. I'm soft selling it, I know, but at the same time, this is one of the only pictures that you as a student or just person in general is going to be able to just, oh no, shit, that's how fucking complicated it is. I really got my shit together if I'm going to start knowing how to do this. So not only does this not even cover the actual cellular pathways, it doesn't even begin to cover the drugs that are going to affect the concepts listed on the wheel. So basically there's an in-clinic drug for each one of those, please excuse that, but basically there's an in-clinic drug basically addressing each one of those points. There are inflammation drugs available. There are angiogenesis drugs available. There are drugs starting to target tumor hypoxia and cytokine signaling. It's usually that the detection doesn't really occur until, at least clinic, somewhere in the cellular to tissue range. But we're getting, the industry is getting extremely good at that with RNA detection, and you get free-floating RNA detection. Just being able to detect a tumor through the use of blood is a major score that never existed 10 years ago. Again, this goes into the wheel of health literacy, where it's like screening and all the wheels are related to each other. So for asset development, it's kind of removed. It's more of my business cycle. But the cancer biology and the health literacy, they're screaming for prevention. There's been huge progress in terms of detection at the biochemical level, not just at the sort of endoscope colonoscopy level, where you need a physical to be able to see it, all the way to cancer smelling dogs. But that goes into how, when I said earlier, this disease is olfactory mediated, but it's a blind eye and a deaf ear kind of thing. That really is where that's where the monoclonal antibody drugs come in. That's where your immunoinhibitors or modulators are coming in from. They're just on the market now. If they have MAB in the name and not the trade name, but in the scientific name, that's a monoclonal antibody that's designed to find one of these to put a nose on a face. Basically, it's a problem. It's the riddle of the Sphinx. How do you put a nose on a face to be able to immunologically be able to identify the cell with the genetic mutation and the altered gene expression? How do you find that needle in a haystack without a nose on your face? And then again, a lot of that, and then that ties us back into the local microenvironment feedback, detection limits, health, and the xenobiotic exposures are all lifestyle mediated. 90% of them are lifestyle mediated. And that's cancer biology in an hour. If you'd like to learn more, so I'm just going to move this over a little bit here. In the corner that I was blocking, it's basically use internet searches in of these terms here, provided to build your knowledge of cancer and cellular biology. So even if you're just like in regular cellular biology, or if you're in, regular if you're in regular biology, if you're in if you're in biology, I'm just gonna say it again. If you're in regular biology, this still applies. You still got to know, how does this fit into your central dogma? Where do your ribosomes kick in? The cool thing on this one is, like, I haven't really paid attention. I haven't looked for it. That's kind of on my this year's to-do list kind of thing. But can you get away with an intratumoral injection of a ribosome poison to stop the protein being expressed and have that as a viable treatment option? You can't do that in a normal method of distribution. You just can't have ribosomal poisons being target molecules for cancer because of everything I just talked about for the last hour. You can't do it because you have all of these healthy cells that are going to take them up preferentially and wipe you out. There are 2 chapters dedicated to this in the audiobook and in the thesis, just sort of explaining how to make a cure, basically. There's stuff you have to consider, there's stuff you have to overcome, but one half of this is already like 30 years into the story. Not that, maybe not that much. Well, we're gonna end here, because that's been the hour, and this has been the heaviest. This is the most complicated one because this is the worst case scenario. Because the worst case scenario is the hardest to deal with. And if you can make an advancement against the worst case scenario, if you can even conceptualize what an advancement looks like, you know this wheel inside out. You might not know all the bits and pieces and the cogs and screws. That's sort of data and science for the sake of data and science. I don't know. I just hope this helps. That's sort of why I'm making these. I hope it helps. I hope it helps explain. If you're going from, it's a little bit 0 to 60, but if you couple that with, your school, your high school, your textbook stuff, your online stuff, your online learning, your independent learning sessions, your regular health literacy, and your PubMed searches, and some of your university level tools that you learn, and Google Scholar to just exist kind of thing. Wikipedia is a good enough place to start for general knowledge. There was a big push earlier that Wikipedia, you don't trust the sources. It's gotten over that for a reasonable amount. You still can't really trust Wikipedia as far as you could throw it, but it's as superficial as this wheel is, and that's enough. That's enough. That's enough. That'll do, pig. That'll do. I'm signing off. I hope you enjoyed this week's Power Hour. A bientôt. Ciao.